Um, Good evening, everyone, and let me at the outset thank Ortho Clinical Diagnostics for asking me to speak in this question answer session and webinar on antibody testing. Before I start, I would like to thank Dr. Krishnan for moderating the session, and the presentation will be brief, and followed by that there are all number of questions that have already been raised, which I will be taking up with the help of the moderator. So can I have my presentation up, please? Amol?
Yes, sir. Can you see the presentation?
No, I
I've put
can't.
it up.
I can't see it. Okay. What I will do now it's come. The outline of this presentation is presented in this slide. I will give a brief introduction of virus structure, antigenicity, and kinetics of responses. I'll be talking on the type of immunological assays. I will also be talking on the current guidelines that prevail in India pertaining to antibody testing, and end probably by talking about the limitations of immunological methods. Next slide, please. You are all probably very familiar with the virus structure and genome organization. I'll take it just a minute. It's a spherical virus. The name is derived from the structure of the virus. It looks like a crown with the radiating projections all around the circumference. That's why it's called corona. Coronaviruses have been known for a long time. 4 coronaviruses have been circulating, causing mild flu-like illness and gastroenteritis for over 50 years. In 2002, we had the severe acute respiratory syndrome coronavirus, SARS-CoV, which appeared in China and spread to various parts of the world. In 2012, we had the MERS coronavirus, Middle East respiratory syndrome coronavirus. And in 2019, December, we had this new novel coronavirus, which is being designated as SARS-CoV-2 because it has some differences in antigen and in genome structure compared to the original SARS virus. So the virus is approximately 60 to 100 nanometers in diameter. It is enveloped. You can see here the blue in the cartoon, the blue circle on the outer side is the envelope. Embedded in the envelope is a very important protein known as spike protein. We'll come to that shortly. And also in the envelope embedded is the envelope protein, which is circled here as a small black one. And then in this envelope and the capsid encloses a single-stranded positive RNA genome, which is surrounded by the nucleoprotein. Now, these three are very important proteins of the virus. We'll come to it shortly. On the right panel, you will see the genome, which is about 27 to 30,000 nucleotides. It has various genes encoding from the 5' end to the 3' end. You will see the open reading frame 1A and 1B. Then you have the structural proteins coming in line. The structural proteins are the nucleoprotein, the spike protein, and the envelope protein, which are there. Next slide, please. In the next slide, you will see the most important protein of the virus, which is the spike protein. This is important for two reasons. Firstly, it is the protein that the virus uses to attach to human cells, especially to the angiotensin converting enzyme 2 receptors that are found on a variety of cells. The spike protein is also the most immunogenic protein. Antibodies that develop to the spike protein are able to inhibit viral entry. That's how we know that it is the ligand for attachment of virus. And secondly, antibodies to the spike protein are protective and they are neutralizing in nature. Next slide, please. The next slide shows you the structure of the spike protein. This is a three-dimensional structure. It has been crystallized and it has a head and a stem. Looks somewhat like the influenza hemagglutinin. Enlarged portion, the boxed item here that you can see is the receptor binding domain, which is marked in this light brown color. The receptor binding domain of SARS-CoV-2 is unique. It is different from SARS-CoV-1, and this is the most immunogenic part of the spike protein. Apart from the spike protein, there are other proteins in the virus, especially the non-structural proteins, which have been circled in red on the right, and they have various immunological functions like modulating the immune response, whether it's interferon signaling or promoting cytokine expression or activating the innate immune system, which I have circled there. Can I have the next slide, please? You all know that there are two arms of the immune system, and both are activated in SARS-CoV-2 infection. The innate arm, as you all know, is the non-specific arm of the immune response. It develops in response to any infection. It is almost instantaneous when the virus infection occurs, and it links to the adaptive immune response. It gives information to the adaptive immune response through secretion of certain molecules known as cytokines and chemokines. The innate immune response does not have memory, whereas the adaptive immune response has memory. That's the biggest difference. The effector cells of the adaptive immune response are T cells and B lymphocytes. The B lymphocytes are responsible for antibody protection, whereas the T lymphocytes are responsible for the cell-mediated immunity. Next slide, please. Moving on to SARS-CoV diagnosis, when an infection occurs, it may or may not result in disease, and when there is disease, it may or may not result in recovery, or it can result in death. So there are a number of diagnostics that are used for various parts of this diagram which I'm showing you to diagnose infection. Globally, it is agreed that the gold standard test is detecting the virus genetic material by reverse transcription polymerase chain reaction. That is the gold standard. Virus isolation can also be done, but it's very cumbersome. It requires a biosafety level 3 laboratory. So it is not practical as a routine diagnostic test. It is more for research applications. Immunological tests have been made available in the last 2-3 months. They are definitely used for epidemiological purposes, but of late there are papers that are being published which say that if you use them intelligently and in combination with RT-PCR, they have diagnostic utility. Next slide, please. This slide shows you the comparison of the different types of COVID-19 tests. The one on the extreme right is the diagnostic RT-PCR test, which is the gold standard. The limitations of this are it is extremely labor intensive. It requires a huge setup of a biosafety level 2 laboratory, a lot of equipments, skill, and it takes anywhere between 6 to 8 hours for a sample to be tested and report generated. However, the advantage is that it's a gold standard test. What about antibody tests? They can be classified into those that are rapid and those that are not so rapid. But these non-rapid tests, there are a number of them. I'll come to them shortly. I'll show you all the different types of tests. But they are far more robust in detecting antibody responses as compared to rapid tests. Next slide, please. Before we go into the actual types of tests and what they detect and how do you interpret results, we should go through the kinetics of responses in COVID-19 infection. This is a graph that is usually used to depict kinetics of any viral infection. There are two axes. One is the x-axis, which is the horizontal axis, which denotes the timeline starting from the time of infection to 1 day, to 4 days, to 5 days, to 15 days, 1 month, 3 months, and beyond. On the y or the vertical axis is the extent to which a parameter is detected or is elevated during infection. What happens in SARS-CoV-2 infection is once the virus enters the human body, it quickly multiplies. It multiplies in the respiratory system. It can also multiply in the gastrointestinal system. But the most relevant specimen for us is the respiratory system. Within a matter of 1 to 5 days, viral titers reach a peak in the nasal and the pharyngeal cavities. So the peak of virus isolation, the peak is around 5 to 7 days, after which the body starts mounting an immune response and the virus level comes down, and it usually comes down in most individuals by 14 days. However, as shown in this graph, virus can be found multiplying in nasal and pharyngeal cavities and in the lung for up to 1 month, depending on how robust the immune response is in the patient. Antibodies start appearing very early, but they reach detection levels only around the 5th day, and depending on the type of antibody, they peak at various stages. For example, the first antibody to appear in SARS-CoV-2 infection is antibodies to the nucleoprotein of the virus, which is shown as a purple or violet dotted line. You can see it starts raising as early as 1 to 2 days. It peaks by around the 7th day, and usually it wanes off by 1 to 3 months. The second antibody to appear is antibodies to the spike protein of the virus, which is the most important protein as it's most immunogenic and it's neutralizing. And the spike protein antibodies appear by around the 5th day. They reach a peak by 14 to 21 days, and after that, depending on the individual, they can fall anywhere between 3 to 6 months, because we have information only up to 3 to 6 months now in the infection. So we know that the antibodies can remain, probably, we do not know how long, maybe, hopefully it will remain for lifelong. So the first antibody to appear is nucleoprotein antibody, and the second antibody to appear is the spike protein antibody. And between these two antibodies, to nucleoprotein and spike protein, we should remember the type or the isotype of antibody also varies. You can have IgM, you can have IgG, and you can have IgA. This graph, however, depicts IgG antibodies, and it also depicts total antibody. There are some assays that detect IgG, IgM, IgA, and total antibody. There are others which detect total antibody and IgG and IgM. We will come to that shortly. Can I have the next slide, please? So depending on the format, antibody tests are divided either as rapid. Almost all of them that are available as rapid tests today are based on the principle of lateral flow immunochromatographic tests, where much like the pregnancy test, if a drop of plasma, serum, or whole blood is put into a well on the device, it flows through by capillary action. I'll show you a picture of it. And when there are antibodies, they meet the antigen, then the conjugate binds, and then you will get a line. That is the lateral flow assays. Other methods that are used are not rapid, like the enzyme-linked immunosorbent assay or the chemiluminescence assay. They also can be done in a matter of an hour or two. The advantages of the non-rapid assays is that you can do large number of samples, whereas in rapid, it is one assay at a time or maybe 5 or 10 assays at a time. Whereas in ELISA and in chemiluminescent assays, you can do large number of samples. The type of antibody detected, I already mentioned, can be IgM, IgG, IgA, or total antibodies. And the antigens used in all these assays have been predominantly the spike protein in most assays. And again, in the spike protein, there are two subunits, subunit 1 and subunit 2. There are advantages of each of them. And people have also used a part of the spike protein, especially the receptor binding domain. Only that has been used as an antigen in either rapid or in ELISA or CLIA tests. People have also used nucleocapsid antigen in ELISA tests. Apart from this, there are other tests which use whole inactivated virus, purified virus coated onto the ELISA plate, and I'll show you an example of that. Next slide, please. This slide, actually, I borrowed from the Ortho people, which very elegantly describes the various types of COVID tests, the sensitivities and the specificities, the quality control issues related to it, the requirement of labor, and the kind of footprint it leaves, the storage efficiency, and the workflow. There are three different tests that have been compared here. Rapid point of care, principle is lateral flow. Where is the setting where we use it? It can be used as a bedside test. It can be used in communities for sero surveys in for rapid testing of large number of populations. It can be used at home, but that licensing for home is still not available. But somebody can go to the home and do it. That is the setting in which. Whereas ELISA and chemiluminescence assays cannot be performed in the field. They have to be done either in a laboratory or in a blood bank. Why do we do it in a blood bank? In a blood bank, we do this test to find out if the antibodies are present to SARS-CoV-2 and if they are present in high levels or with high OD. Those donors or the people who have high levels of antibodies can serve as potential donors for plasma exchange therapy, which we'll come to later. The choice of these tests will depend on the setting. If the setting is a clinic with small number of patients, then rapid tests, whereas for wider use, both ELISA and CLIA can be used. The type of results generated in a rapid test are only qualitative, positive or negative, whereas ELISAs and chemiluminescent assays can be both qualitative as well as semi-quantitative to give you an indication if the level of antibody is high or it is low. We do not have still assays that can measure antibodies in units. I'm sure it will come up. The ELISA and chemiluminescent can measure IgG, IgM, IgA as well as total antibody, whereas the rapid test can measure only IgG and IgM. The specificity and sensitivity of ELISA and chemiluminescent assays are far, far higher compared to the rapid tests. And quality control can be controlled with the ELISA and chemiluminescent tests. The tests require equipment and skill. They leave a medium or a high footprint, especially the CLIA assay, and workflow can be automated in both ELISA and chemiluminescent assay, whereas it is manual in rapid assays. Next slide, please. This actually summarizes what I have mentioned in the previous 3 slides. Antibodies are developed to various antigens. They can be IgM or IgG, and IgM is generally a marker of early infection, and IgG appears later than IgM, and usually IgG means that the person is beyond 14 days of infection, generally speaking. And the sensitivity and the specificity, as I said, are higher for the ELISA and the CLIA. Next slide, please. Okay, and this slide shows you exactly how the tests are done. I was telling you how the rapid test works. You put the specimen drop in a well, it flows through capillary action, then it meets a line, a dam where antigens are kept, and then anti-human IgG or IgM is kept. And when it meets that, the antibody stops, the streptavidin conjugate binds, and you will get a red line. So this is a test which can tell if antibodies are present or not. The most important test from the point of view of assessing protection, whether it is to give an immunity passport to an individual or for vaccine efficacy testing, is the neutralization test. It's a very cumbersome test. You require live virus, you require biosafety level 3 lab, you need to dilute serum and incubate it with live virus, various dilutions, then put it on a monolayer of cells and overlay it with agar after the virus attaches to the cells. Then you will see clear plaques in these plates. This is count the number of plaques and see how many plaques are reduced in the presence of antibody as compared to no antibody. This is called plaque reduction neutralization test. This is the gold standard for measuring protective immunity. ELISA, chemiluminescent, and all the other assays that are used are basically where antigen is coated on a solid matrix. You react it with the patient's sample and then wash it and then come up with a labeled antibody. If the labeled antibody has an enzyme attached, it is ELISA. The labeled antibody has a chemiluminescent compound attached, it is called chemiluminescent assay. If the labeled antibody has fluorescence molecule attached, then it becomes an immunofluorescence assay. Next slide, please. Let's move on to what the government of India is telling about use of antibody tests. This is one of the first notifications issued by the Indian Council of Medical Research, which actually provides rules and guidelines in the country for use of various tests. As you can see from this communication, the Indian Council of Medical Research has evaluated a large number of rapid tests, and in this test you will see, in this notification you will see what are the recommended kits. But come down to the right bottom, the guideline or the recommendation of ICMR is to be followed in toto. Rapid antibody tests can be done on blood samples and plasma samples. Test results are available within 30 minutes. test can be positive 7 to 10 days after infection, and this is not to be used as a diagnostic test, meaning based on the test results, you cannot say somebody is currently having SARS-CoV-2 infection. A positive result only means somebody had been infected with SARS-CoV-2. A negative result, most importantly, does not rule out SARS-CoV-2 infection. Next slide, please. Subsequent to issue of this guideline, and here is an example of a rapid test. I told you in the device, in the well, you add a serum or plasma or whole blood, depending on the kit. Then you drop as shown on the left side, a buffer, a few drops you add that drives the blood into the device. And when it meets IgG or IgM, then you will get a red line. Various interpretations are shown. But one condition for the test is the control line, or what is marked as C, should come positive. Next slide, please. Here is an example of an ELISA test, which is developed by, next slide, please, by National Institute of Virology. It is already available in the market. It detects antibody to inactivated whole virus. It is marketed by the name COVID Kavach. This test has been compared and with the neutralization test, and it has high correlation. So antibodies detected in this test are a surrogate marker indicating that the antibodies are probably neutralizing. This test takes approximately about 2 hours to complete, and this kit is available in 90 well format already in the market. Next slide, please. Continuing with the ICMR advisory on antibody testing, it very clearly says IgG antibody detection for SARS-CoV-2 is useful in the following two situations. One is to conduct sero surveys. Why do we conduct sero surveys? We want to know the prevalence of SARS-CoV infection in a community, most maybe a containment zone, or maybe among healthcare workers, maybe among frontline COVID workers. Then you can employ antibody tests, either rapid or ELISA or CLIA or immunofluorescence. Especially many people in India are now using antibody tests for surveying high-risk vulnerable populations, especially in hospitals, because many hospital workers are afraid that they have already contracted infection, or they want to know their sero status. So in such situations, ICMR definitely recommends it. Next slide, please. However, to carry out antibody tests, ICMR is mandated to review and conduct research on evolving trends of the disease, accordingly advise the states, country on the public health policies. The ICMR has said that the respective state governments have to give permission for doing this test, or they should keep the state informed that they're doing the antibody test. But most importantly, all the results that are generated in any antibody test, a comprehensive report has to be submitted to ICMR to the email address mmurekar@gmail.com. ICMR is collating all sero survey results and antibody test results so that it has an idea of how the prevalence of the infection is in the country. So whoever does antibody tests, they definitely have to share their results periodically with Dr. Manoj Murekar, Director of National Institute of Epidemiology in Chennai. Next slide, please. You can skip this and go to the next slide. You can skip this. The same thing what I mentioned is summarized here. Next slide, please. The CDC also has provided advice on COVID antibody testing. Especially it says people who receive positive results of an antibody test but don't have symptoms or have not been around someone who may have COVID are not likely to have a current infection. They can continue with normal activities, including work, but they should take steps to protect themselves and others. It does not mean having antibodies means you can go around freely and you are protected and you are you're not going to get the infection, nor are you going to transmit the infection. It depends on when in the window or in the kinetics curve that I showed you, there are later on also I show you there are some times when antibody is positive and RT-PCR is also positive. So you can still transmit infection. Antibody detection by any of the methods does other than neutralization test does not mean you are protected. That is the warning. And it also says people who have a positive result should care for themselves and be responsible in not transmitting infection to others. So, this is an important point I want to underscore. Next slide, please. A very useful publication for those who are interested. It's kind of summarizes and it's a review in clinical microbiology and infection, which talks about diagnostic strategies for SARS-CoV-2 infection and more importantly, the interpretation of microbiological results. At this point, I want to mention now antigen detection tests are also available. Now, many institutions are very enthusiastic in using RT-PCR, antigen detection, and antibody detection. Reading this paper will give you a clear picture on when to use what and how to interpret results. Next slide, please. This is a paper that is published a few days back by the Harvard group that has compared RT-PCR results and total antibody results in a same set of patients. If I'm not mistaken, I think it was 209 patients, and among those 157 where RT-PCR was done and 157 also had antibody results. Again, this graph, let me explain. X-axis is days after symptom onset, starting from 0 day to 38th day, and on the y-axis or the vertical axis is the sensitivity or the percentage positivity that you had in these patients. As you can see, in the first 5 to 7 days, RT-PCR has a very high sensitivity. What do you mean by sensitivity? The pickup rate in RT-PCR is to the tune of 95% on the third or fourth day. After the seventh day, you can see the yellow curve dropping down, and usually RT-PCR becomes negative in a majority of the individuals by 14th day, and in approximately up to 20% of the people, it can be positive up to the 4th week of illness and sometimes even beyond. On the other hand, antibody detection is not so very efficient in the first 3-4 days. It starts picking up by the 4th, 5th day, reaches a peak definitely by the 14th day, and by 20th day, 100% of the people are likely to have antibodies. It may be IgG or IgM or IgA. I'm talking of total antibodies. At day 9, 10, or between day 8 or day 7 and day 14, both of them intersect. I was making a point to you on the advisory from CDC. At this stage, remember, antibodies are positive. Nucleic acid or RT-PCR is also positive. So the person can be infectious even with the presence of antibody in their serum. It's only beyond 4 weeks you see antibody is negative and nucleic acid or virus genetic material is negative, whereas antibody is positive towards the far right of this curve, telling you at that time you are not infectious. Next slide, please. The same paper also talks about the clinical interpretation value of the various diagnostic tests compared to symptoms. If symptoms are present, RT-PCR is positive and antibodies are either detected or not detected. The interpretation is it is acute infection. If symptoms are definitely positive, RT-PCR is negative. Antibodies, whether IgG, IgM, or the spike protein or nucleoprotein is present, that means it is recent infection. If symptoms are present and antibodies are present, all types of antibodies to all antigens, then probably it is late onset infection. If RT-PCR is negative, symptoms are negative, only antibodies are positive, it is old infection. This person is no longer infectious. If all of them are negative, then it is absence of infection. Next slide, please. Okay, before I end, I thought I will just give you a sense of the Vitrios anti-SARS-CoV-2 antibody, which the Ortho Clinical Diagnostics company has made. It basically detects antibodies to the spike protein of SARS-CoV. It uses a combination of biochelated recombinant SARS-CoV-2 antigen, which binds with the antibody in the patient sample, and then you can detect it using a chemiluminescent signal. Okay, and the Vitrios test was evaluated for interference with bilirubin, with hemoglobin, and with intralipid, and they have shown that there is no interference even in the presence of high levels of these biochemical reactants in patient sample. Next slide, please. The assay has been validated by Mayo Clinic laboratories, and they have said that it is 100% sensitive, whether it is at 1% prevalence in the population or in populations that have higher prevalence. Sensitivity is not compromised, it is 100%. Specificity is over 99%. The positive predictive value is very, very high when we are using it in a setting of 5% prevalence, which is what most of the countries are now. At least the major countries are now at prevalence of 5%. Next slide, please. What is more important is in the Mayo Clinic evaluation, what they have found when they did serial samples collection from COVID-19 RT-PCR confirmed hospitalized patients, they found that by day 14, the orthoclinical CLIA assay was able to pick up antibodies, IgG antibodies, in all 100% of the patients. Next slide, please. The slide is not visible, Anmol, very clearly. The next slide. Can you move forward, Anmol?
Sir, has it come up?
Yes. Before I end, I will talk to you about next one. Yeah. Okay. Before that, I will tell you what is the role of serology. The previous one, Anmol. To summarize, then I'm coming towards the end of my presentation. There are the role of serology or detection of antibody. What is it today? It is used in serosurveys, especially in epidemiology, to estimate prevalence. It is very importantly used to detect potential plasma donors. As you know, plasma exchange therapy is one of the methods people are using. The third scenario where antibodies are used is to estimate antibody responses following vaccination. As you know, a number of vaccine candidates are already entered phase 1 and 2, and one of them has also gone to phase 3 trials. So there, the only marker available to estimate potency and efficacy of vaccines is to measure antibodies, and ELISA and CLIA are being used for that. The antibody assays, most national and international agencies are not recommending it as a standalone for diagnostic use, although literature is now piling up saying that in combination with RT-PCR, if you use it intelligently, then you can have certain amount of diagnostic interpretation that can be given to antibody assays. Next slide, please. There are also limitations with antibody detection assays. I will probably end with this. What we know in the last 5 months is, unlike many other acute viral infections like dengue, chikungunya, Japanese encephalitis, influenza, and so many others, SARS-CoV-2 responses are not as brisk and as early as other RNA viral infections. It takes 7 days for the initial response to be detected and hits a peak by 14 days. I can give you an example of dengue or Japanese encephalitis. It peaks much earlier. It peaks by 7th, 8th day, and by 10th day, almost all patients have antibodies to the other RNA viruses. We don't know why. We are learning. Hopefully, we'll find an answer. There is one study that has reported from Mount Sinai that all 100% of patients do not develop antibodies. I'll show you the data. One general remark, rapid antibody tests sensitivity is not what most kits are claiming. They have not lived up to the promise of their claims. I'm talking of whatever little valuations we have done, about half a dozen rapid kits. None of them have lived up to their claim of 90% at 14 days. Neutralization assays are the gold standard for protection, but they are very cumbersome and very technically demanding, and high level of biosafety is required. In the next slide, I will show you the study from Mount Sinai Hospital. Can you go to the next slide, please? This is published on 5th May 2020. It shows that humoral immune response is not found in a very small percentage of people. There are two graphs here, A and B. A graph is the number of the titer of antibody measured in an ELISA. As you can see, in the number of patients that they have done, I think this is more than 300 patients, if I'm not mistaken. 11% did not have antibodies at all at the end of 14 days. So they went back to these patients and collected samples again at 31 days. From the original 11%, 90% developed antibodies by 30th day, but still there were 11% beyond the 30th day who did not have antibodies. So there is a small proportion of people who are not seroconverting in terms of antibody. Probably they are mounting a cell-mediated immune response. So one should keep in mind that some patients may not develop antibodies even at the end of 30 days. I think that's the last slide, Anmol. Can you go to the slide? Thank you very much. You've been a very patient audience. I will now have time for questions.
Thank you so much, Dr. Ravi. It was a very loose presentation. To quickly go through some questions that I have here from the participants. The first question is about from Dr. Darshan. Any lab can do IgG antibody. The question is, can any lab do IgG antibody, or only NABL, CAP, or ILAC labs can do it? What is your comment?
There is no clear-cut instruction from ICMR on it. So I presume any lab can do it. There are people who approached me in Karnataka, and I am sending letters to them that they can go ahead. The condition, however, is that antibody results have to be sent to the email address I showed, mmorekar@gmail.com. Whatever is the result, in the form of a line list, they can give the details of the patients. They can anonymize and they can give a lab ID, the, the gender and the age, and what was the result, and what kit was used, so that the ICMR will have the information as a national database for analysis.
Thank you so much, Doctor. The next question is from the same person. Which are the necessary PPE one should wear while performing CLIA in the laboratory?
Actually, there is no need to wear PPE for serological tests. Masks and gloves and aprons are enough. The amount of virus that is found in the serum samples is very, very less. It is not recommended that you need to use PPE for serological testing. The only situation where you will use PPE for serological testing is in the field when you're collecting sample and doing a rapid immunochromatographic test, because you are at that time with the patient and collecting the sample.
Right. Okay. Thank you, Doctor. The next question is about resource constrained situations where RT-PCR is not set up. Say, for example, tier 2, tier 3 cities. So the question is, if only IgG test is done, and if the test returns positive, is there a necessity to do a confirmation by RT-PCR for checking whether there is infection or not?
There are two situations I would like to mention. First is this person a known contact? If the person is a known contact and turned IgG positive, how many days after contact was serum sample collected? You remember the crisscrossing graph I showed on how RT-PCR goes down, being very highly positive in the first week up to fourth week, and antibodies start going up. So if we know the contact history, the duration of contact, I would say the person in this graph that is up on the screen between the 7th and 14th day, I would still do an RT-PCR to know if he is currently infected or not. The second scenarios, we do not know whether the person had any contact, but he is antibody positive. If he feels that he wants to rule out infection, he can go for a swab test because he does not know any contact to satisfy himself. So I think antibody tests have to be interpreted very cautiously in relation to whether there is active infection currently or not, and for that you definitely will need RT-PCR.
Thank you, Doctor. The next one is about the utility of the antibody test, especially the total antibody test. Is it advisable to introduce the total antibody test anti-SARS-CoV-2 as a part of pre-operative screening for all the patients, as well as for all those CKD patients who are undergoing dialysis?
This is again a very tricky question. If antibodies are positive, don't rush saying that this person is protected so I can operate on them. There is no replacement for taking precautions. Antibody test does not tell you that you need not take precautions. The golden rule is everybody is COVID infected unless proven otherwise. Okay? If you want to take decisions on whether to operate or not, antibody test is a screening test. If it is positive, then you'll have to ask the question, how long has this patient been in contact with somebody? Or case 2 is, I don't know whether this patient was in contact with anybody. RT-PCR is needed. In a negative scenario, anyway RT-PCR is needed. This is a call that the treating surgeon or the team has to take. Antibody test is not a replacement for RT-PCR. I underline that.
Okay. That's a very clear statement, Doctor. The next question is about the reporting part to ICMR. The question is, should we enter the report into the ICMR portal? And before that, should we register in any portal for starting the test for ICMR?
ICMR has made it very clear that there is no registration in the portal required. The portal is for, as of now, primarily for RT-PCR. That is the portal meant for. If they are doing antigen tests, they have to directly get in touch with ICMR and they will tell you how to enter the antigen results. As far as antibody results, ICMR guideline is very clear. You should use only ICMR approved kits for doing the test. And once you have the result in a line list format, specimen number, age, gender, location, we are from which part of India, and what assay you use, what is the result of the assay. This may be once a week. You go on updating your line list and sending it to Dr. Manoj Murekar. There is no separate registration, but you will have to inform the local state authorities that you are starting and get an okay from them.
Okay, just the last couple of questions, Doctor. You talked about there are tests available for individual antibody detection, IgM, IgA, and IgG. So the first question is, what is the significance of IgA alone test or IgM alone test? What is their significance?
IgM alone, the utility is, it is a marker of acute infection. It's the first antibody generally in any viral infection to appear, but there are papers in SARS-CoV saying that it may not necessarily be so in SARS-CoV-2. Many people have IgG appearing before IgM. There are publications supporting that. That may be related to prior exposure to other other coronaviruses. That's one of the reasons, especially if cross-reactive antigens have been used. So one has to keep that in mind. But IgM, as I showed you very clearly in a table of interpretation, Anmol, you can bring it up. If only IgM is positive, all the others are negative, then it probably means recent infection. But such a situation is usually rare. From whatever I have reviewed publications, only having IgM is very rare, because IgG appears for some reason in SARS-CoV-2 as early as IgM. We do not know why the kinetics is like this in SARS-CoV-2. As far as IgA, there are people who are pushing for IgA, saying that it's a diagnostic marker. It comes much earlier than IgG, and they are also pushing it as a home test so that you can put a pad or a device into the mouth and pick up IgA antibodies in saliva. But all these are preliminary observations. Rigorous validations have not been conducted to say that it has utility as an early marker of antibody response. Over.
Okay. The last question, Doctor. For the SARS-CoV-2 virus neutralization, is only IgG antibody effective for neutralizing the virus, or what is the role of IgM and IgA in virus neutralization?
Let me tell you, isotypes cannot be linked to functional aspect. Generally speaking, IgG antibodies are neutralizing, but there can be IgM neutralizing antibodies, and there are also IgA neutralizing antibodies. In the neutralization assay, we do not type the immunoglobulin. We are looking only whether among all the antibodies that are present, whether there is protection or no protection. It is not linked to A, G, or M. So I want to make that point very clear. Most often, if you look at experience with other viral infections, it is IgG that is neutralizing. Why? IgG is a marker of immune maturation. As we go on into the time course of an illness, the immune system matures itself and produces antibodies that fit the virus much better and therefore are capable of neutralizing. But there are instances where M and A also have been shown as neutralizing antibodies. I hope I answered the question.
Yeah, yes, doctor. Doctor, thank you so much for the patience and the very clear answers that you've given for all the questions. Thank you so much. Over to Amol for closing.
Thank you, Dr. Ravi and Dr. Krishnan, for a very clear presentation and taking up all the questions. And thank you very much for all the attendees for sharing your time on these webinar series. Maybe there would be a few more questions coming which we would not have picked up. We would reach out to you individually once we get the response from Dr. Ravi. Thank you very much, and we will meet tomorrow for another webinar series.
Thank you very much to Ortho for organizing this. Have a nice evening.
Thank you, sir.